Hello everyone. Today we will discuss about inborn errors of metabolism. Inborn errors of metabolism are a large group of rare genetic diseases that generally result from a defect in an enzyme or transport protein which results in a block in a metabolic pathway. So if any defect in these enzymes, there will be accumulation of substrates. These substrates, either they are excreted in urine or their level will be increased in the blood. Some of these accumulated substrates will become toxic. So these substrates will take alternate pathway. So instead of normal products, there will be abnormal byproducts. These abnormal byproducts are toxic. There will be elevation in blood or they will be excreted in urine and can detect in the urine. Coming to the classification of inborn error of metabolism, there are different categories like errors of carbohydrate metabolism, errors of amino acid metabolism, lipid metabolism, errors of nucleic acid metabolism, disorders of heme metabolism, lysosomal storage disorders, peroxisomal disorders, trace metal disorders, etc. First one, errors of carbohydrate metabolism. In this, glycogen storage disorders, type 1, von Jerk's disease, due to the deficiency of enzyme glucose 6-phosphatase. Type 2, Pompe's disease, enzyme deficiency is lysosomal alpha-1,4 glucosidase. Type 3, Cory's disease, debranching enzyme. Type 4, Anderson's disease, branching enzyme. Type 5, McArdle's disease, muscle glycogen phosphorylase. Hirsch's disease, liver glycogen phosphorylase. Seventh one, Tarwe's disease, phosphofructokinase enzyme deficiency. Next one is fructosuria due to the deficiency of enzyme fructokinase, hereditary fructose intolerance, aldolase B, galactosemia, enzyme deficiency is galactose 1-phosphate uridyl transferase, lactose intolerance, enzyme is beta galactosidase, then diabetes mellitus, mucopolysaccharidosis, G6PD deficiency. So these are the inborn errors of carbohydrate metabolism. Coming to the errors of lipid metabolism, this will be of three categories: disorders of fatty acid metabolism, lipid storage disorders, and disorders of lipoprotein metabolism. First one is fatty acid metabolism. This includes carnitine transporter deficiency, carnitine palmitoyl transferase 1 deficiency, carnitine palmitoyl transferase 2 deficiency, carnitine acyl carnitine translocase deficiency. Second one is lipid storage disorders. This include Tay-Sachs disease, deficiency of the enzyme hexosaminidase A, Gaucher's disease due to the deficiency of enzyme beta-glucosidase or glucocerebrosidase, Niemann-Pick disease, sphingomyelase enzyme, metachromatic leukodystrophy, aryl sulfatase A, then Fabry's disease, galactosidase, and Krabbe's disease, galactosyl ceramidase enzyme deficiency. Next one is disorders of lipoprotein metabolism. This includes hyperlipoproteinemias and hypolipoproteinemias. Hyperlipoproteinemias are divided into primary and secondary. Primary are genetic disorders. Secondary are due to underlying disease processes. Primary is of 5 types. Type 1, familial lipoprotein lipase deficiency. Type 2, familial hypercholesterolemia. Type 3, familial dys-beta lipoproteinemia. Type 4, familial hypertriglyceridemia. Type 5, combined hyperlipidemia. And this hypolipoproteinemia is of 2 types, A-beta lipoproteinemia and familial alpha lipoprotein deficiency. So that is inborn errors of lipid metabolism. Coming to amino acid metabolism disorders, it includes amino acidopathies, organic acidopathies, and urea cycle defects. For example, phenylketonuria, here the defect of enzyme phenylalanine hydroxylase, hypertyrosinemia, fumaryl acetoacetate enzyme, alkaptonuria, enzyme deficiencies, homogenesic acid oxidase, homocystinuria type 1, cystathione beta synthetase, type 2, methyl transferase, type 3, methylene THFA reductase. Then histidinemia, enzyme defect is histidase, maple syrup urine disease, branched chain keto acid decarboxylase, methyl malonic aciduria, methyl malonyl CoA mutase, cystathionuria, cystathionase, hyperprolinemia, proline dehydrogenase, citrullinemia, arginosuccinate synthetase, arginemia, arginase, hyperornithinemia, ornithine decarboxylase, then ornithine transcarbamylase deficiency, carbamide phosphate synthetase 1 deficiency, then arginosuccinate aciduria due to arginosuccinate lyase deficiency. So these are the inborn errors of amino acid metabolism. Errors of heme metabolism include porphyrias, Crigler-Najjar syndrome, etc. Trace metal disorders include Menkes-Kinckey hair syndrome, Wilson's disease, etc. Peroxisomal disorders include Zellweger syndrome, adrenoleukodystrophy, etc. So these are the inborn errors of metabolism. This is today's topic. Thank you for watching.